Hi everyone. So my name is Christopher Xenakis. Hopefully you guys are doing well in the class for biochemistry. Um, and today is November 12th, 2021. And this is my presentation for the Unit 2 assignment. So from the following four Unit 2 cases, I chose to do case 2 and number 4. So I'll start with case 2. I'm not going to repeat the whole question passage word for word. So just to explain briefly, we have a female in her mid-teens that was diagnosed with a muscle glycogen dephosphorylase. I'm sorry, a muscle glycogen phosphorylase deficiency, and we need to explain to her family, in layman's terms, what the condition the daughter has, what kinds of actions the condition prevents the body from doing, and if she can basically still lead a normal life. First, I'll explain what the normal role glycogen plays during exercise. So, glycogen's main role in the body is to act as a type of energy storage that gets deposited in our tissues and liver primarily. And in order to synthesize glycogen, we need the help of enzymes, which are proteins that help speed up reactions in the body to assist in metabolism, which is also how we break down or build up substances. If I say a word that ends in ACE, you'll know that I'm talking about an enzyme. With the help of the enzymes, phosphoglucomutase, UDP, glucose pyrophosphorylase, and glycogen synthase, we can convert glucose 6 phosphate all the way to glycogen, which is our end product. If we want to break down our glycogen back into free glucose, we'll use different enzymes called glycogen phosphorylase and glucose 6 phosphatase so that it can be released from the liver. Now, just like in normal life, we never want too much or too little of something. So this needs to be regulated. And for skeletal muscle, which is what we are dealing with in this patient's case, the body regulates by monitoring the activation of epinephrine, AMP, or adenosine monophosphate, and calcium. The enzyme I mentioned before, glucose 6 phosphatase, will not be present in the skeletal muscle since that deals with glycogenolysis in the liver. Now, during exercise, we do continuously deplete our glycogen storage supply as the body requires it after prolonged periods of running, weightlifting, etc. Without enough glycogen, it can lead to fatigue and low stamina. And with low amounts of muscle glycogen phosphorylase, aka McArdle's disease, which is a type of glycogen storage disease, glycogen 1 phosphate won't be able to be released from glycogen, which leaves glycogen to accumulate in the tissues, which can then lead to poor exercise tolerance. To answer the family's question, no, she will not be able to take long walks since she will most likely develop muscle cramps and quick fatigue after just a short while. Someone with this condition also needs to watch their food intake, meaning to manage one's consumption of carbohydrates, creatine supplements, as well as consuming sucrose before performing any type of exercise. Now, the next question about having to get up at night to eat to maintain her blood glucose levels. The answer is no, because the purpose of this condition is the body isn't able to utilize glycogen for energy, which leads to an accumulation, which I mentioned before. So we don't want to exacerbate this by having her eat more at night. And in addition to that, the family also wanted to know if she has to worry about producing excessive lactate during intense anaerobic exercise. So to answer this, she should be slightly worried just because the goal of anaerobic exercise is to expend quickly and immediately any type of energy. So that would involve like heavy weightlifting, HIIT training, or which is high intensity interval training, sprinting as well, and so on. So not even jogging, just mainly just sprinting for very short distances or long distances, but very quickly. So if she's going to be doing these exercises, lactate would be produced exponentially, since pyruvate would convert to lactate when oxygen is absent. In addition, she should try to avoid these high intensity exercises that come with a high expenditure of energy and resort to exercises that she can do at a normal pace that can be separated by 10 to 15 minutes, let's say, based on her condition. So now I'll be discussing case 4. So I want to briefly go over the question where they're basically telling us we have a 2-month-old female with jittery spells that occur after breastfeeding and hypoglycemia as well. Epinephrine was administered and there was no increase in the blood glucose, but there was an increase in blood lactate. She was also confirmed to have hepatomegaly and her glucose did go up after breastfeeding, but she was able to maintain those levels within a normal range during fasting. So we have to figure out which of the 4 diagnoses here are the most likely. So I'm just going to go from A through D. So I'll start with A, which is hereditary fructose intolerance. So this condition is associated with a lack of aldolase B, which is an enzyme that cleaves fructose 1-phosphate into DHAP and glyceraldehyde. So without aldolase B, fructose 1-phosphate would get trapped in the liver, causing hepatomegaly, which is one of our clinical features, as well as fasting hypoglycemia, which is also one of the clinical features. There would be reduced sugar or fructose in the urine, vomiting, irritability after eating fructose. We can already eliminate this choice since the passage doesn't really mention the baby consuming fructose, only breast milk, and the accompanying symptoms really can be present in other diseases as well. So let's move on to choice B, which is galactosemia. So this disease is characterized by deficiency in galactose 1-phosphate uridyl transferase, which in turn blocks galactose metabolism and inhibits hepatic glycogen metabolism and other pathways that require UDP sugars. We can assume that this is the answer since one of the main clinical features of galactosemia is damage or abnormal changes to the liver, which the patient does, but the condition also emphasizes jaundice and cataracts, which the 2-month-old didn't exhibit, and it would be important to note for this case because those are the 2 main clinical features. In addition to that, the passage also mentioned that the baby had an increase in blood lactate after epinephrine was administered, indicating that gluconeogenesis was able to take place. So we can pretty much eliminate B. So now we'll move on to C, which is glycogen storage disease or the inability to use glycogen. Or GSDs for the acronym. GSDs basically are subdivided into many different types depending on the enzyme that's missing. So their goal is to alter or mutate a specific gene in order to block the production of the enzyme that breaks down glycogen. It can allow glycogen to build up in the liver or just not have it produced at all. And symptoms of GSDs do include hepatomegaly and hypoglycemia, which the infant is exhibiting for both, as well as the inability to maintain glucose levels, which is the main cause of concern for the infant right now. So this seems to be the most likely diagnosis, but let's move on to choice D to confirm. So choice D, which is glucose 6-phosphate dehydrogenase deficiency, is characterized as a disorder that's associated with the pentose phosphate pathway and red blood cells. So glucose 6-phosphate dehydrogenase is one of 2 required enzymes for the irreversible oxidation portion of the pentose phosphate pathway that helps convert glucose 6-phosphate to ribulose 5-phosphate. So by losing this enzyme, NADPH would decrease in red blood cells that causes increased cellular oxidation, hemolytic anemia, which is when red blood cells are destroyed faster than they are produced, and jaundice as well. So this does not relate to the infant's condition at all, since lab results only revealed hypoglycemia, but other than that, nothing remarkable. So low blood glucose, but nothing else other than that. So we would have seen a decrease in red blood cell count if we did labs for a complete blood count, obviously, which the doctor should have done, but he said nothing remarkable regarding that. So we can eliminate this choice. So this leaves us with choice C. The patient most likely has this diagnosis. So this is a type of the preferred type of glycogen storage disease for this patient would probably be type 3A or 3B, which indicates a lack in the liver and or muscle debranching enzyme. So that's about it. I'm going to attach the references up to the class. Thank you so much for listening, and hopefully everyone's doing well.